Okay, so when we last left off, we are on the mitosis phase of cell division. So we've already made it through the whole interphase, which consisted of the G1, which was just the growth happening, the beginning of the replication of the DNA, and then it went into the S and then the G2. And we talked about different stop points where the cell was checking to make sure that everything was going correctly before it continued on and actually completed the cell division. So they didn't want any mutations to occur. So the next phase is the mitosis. There's actually a video for you to watch. Make sure you watch that. It's an Amoeba Sisters. So, but we're going to continue on the notes for today about mitosis. And that's what we're going to do. So my suggestion would be if you need to write something down, just pause the video, write, and then press play as you go. Okay, so here we go. Okay, so this process sorts and divides the duplicated chromosomes, because at this point, remember in the G2, we saw the duplication happening. I think it was in the S phase, actually. The duplication had already occurred, and then it was starting to divide. So that's what the mitosis does. It sorts and divides those duplicated chromosomes. We went from one set, and then we split. And now they're starting to sort them and divide them into two identical sets of DNA so that each daughter cell has its one set. Okay, so here we have the parent cell, we have our DNA. And then during the S phase, remember, it duplicates them and it duplicates the centricles. And mitosis is needed and it sorts those chromosomes out. Okay, so that each daughter cell gets the exact as what the parent cell had originally. So your daughter cell ends up looking like your parent cell. Okay, correctly sorting them. However, mitosis isn't always perfect. Do you think there's another stop point? Let's hope so. It says, sorry, my thing is in the way. There we go. Two non-identical sets of chromosomes in daughter cells. Oh, do you see how there's a difference here? Oops. Is incorrect sorting, and an error in mitosis has occurred. So you can see that it should have a red, blue, and a green. And instead we have two blues and a red, two greens and a red. Okay, so there's been an error. So humans have 46 chromosomes in their somatic cells. So here you can see karyotic type showing 46 chromosomes for the human. This means that at some point in mitosis, 92 chromosomes must be sorted and separated. Okay, and that's just because you're doubling that 46 to get 92. Mitosis is broken down into 4 main stages. So again, that interphase, remember the biggest section of cell division has 3. Mitosis actually has 4, and they can be represented by the acronym PMAT, which stands for, boy, that's not as easy as just remembering KFC for dividing, right? PMAT. It stands for the prophase, metaphase, anaphase, and telophase. So I think there's a spot for you to also write that in your pie chart of your cell division. It says, however, the events that occur between the prophase and the metaphase are important enough to create a 5th stage between the two called a prometaphase. Maybe that's the checkpoint. After mitosis divides the nuclear content into 2 separate times, it's time to divide the cytoplasm. Everybody remembers what the cytoplasm is? The cytoplasm gels. Cytokinesis is the very last of the process. And the process that divides the cytoplasm into half to create 2 separate daughter cells. Animal cells is called a cleavage furrow. They form to constrict the cell membrane in the middle of the cell and divide it. So it looks like that. You can see like this furrow that has began, this cleavage furrow that helps to divide. That's in all animal cells. Plant cell looks a little bit differently. Again, here's another picture of it. In plant cells, the cell wall makes the cytokinesis more challenging. Instead of making a cleavage furrow, a plant makes a cell plate in the middle of the cell. So you can see the plate beginning in the middle and then begins to block and split the two cells. The cell plate is made up of vesicles, which contain the material needed to build a cell wall. And the vesicles fuse at the midline of the cell to create a new cell wall, which completely separates the two daughter cells. Okay, so now we're going to look a little more closely at mitosis. So we have, remember that part of the interphase, the G1, which is a big section of, this is mitosis, interphase. In this example, we'll examine a cell containing 3 chromosomes, a red, blue, and a green, an initial parent cell. And then again, the G2. After the S phase, each chromosome has undergone DNA replication, and now we've doubled, right? So now there are 3 duplicated chromosomes. At this point, the DNA has not condensed yet, so it will still be long and thin. This is like a thread-like form of DNA, is also called chromatin. Again, the interphase, remember that's the longest time of the cell. The two identical sides of each duplicated chromosomes are called sister chromoids. So here they are. And they are connected together at a region of the chromosomes called the centromere. And you can see that there's a connection right here. They stay connected at the beginning. Okay, and you can see this connection that's occurred. It's the site of the attachment and the microtubulars, which pull the sister chromatids apart. Okay. Microtubes are protein filaments that serve as various different functions within the cell. They are used in mitosis or meiosis. They can be specifically referred to as spindle fibers. Okay. In the G2 interphase, those pairs, along with proteins that surround to form it, those are our centriole pairs. Remember, those also have duplicated in this process, where it is responsible for organizing the microtubes. And these are only found in animal cells. Along with the DNA, it is also replicated in the S phase to produce 2 more, like you see in the G2. And then here come the 4/5 phases of mitosis. So remember, we have interphase first, which is the longest section of cell division. And now we come into the mitosis section. And that's after everything has been duplicated. Okay, so here comes our prophase. It's the first stage of mitosis. And they begin to migrate. So what they're going to do is they're going to start to move to opposite sides of the cell, opposite poles. So you start to get one that's on one side and one on the other side. This is part of prophase. And the spindle fibers begin to form between 2. So they're also going to begin to kind of like migrate. The nuclear cell membrane also begins to dissolve. So what had been protecting them, that membrane, begins to dissolve. Why do you think the nuclear membrane needs to dissolve completely? What do you think? Okay. This frees up the chromosomes to be sorted and divided in later stages of mitosis. So you have to get rid of the membrane so they're able to move around better and separate. The chromatin begins to condense and become shorter, thicker, and more visible. So remember, before these were more string-like, more like a spindle looking, and they begin to condense and kind of tighten up. They become shorter, thicker, and more visible. and the nucleus begins to disappear. Okay. This is the next stage, the prometaphase. And the nuclear membrane is almost completely dissolved, which allows the spindle fibers access to the duplicated chromosomes. Okay. And the nucleus has disappeared. Okay. And the spindle fibers reach through the dissolving nuclear membrane and attach to these pieces right here, each duplicated chromosomes. And the spindle fibers begin to position the duplicated chromosomes along the middle of the cell, called the equatorial. Maybe because it has something to do with, let's see, equal in the middle. All right. And there's that plate. Probably because it takes it and splits it right in half equally. That would be my guess. Equatorial plate. Okay. And the polar spindles from opposite poles reach the midline and push against one another to extend the distance between the two centrochromes. Okay, and they help to anchor them on opposite sides of the cell. So we have all these little spindle fibers that are pulling and moving things around to help it to divide and equally get each thing on each side. Okay, a spindle apparatus. So different types of spindle fibers help form a network of fibers called the spindle apparatus. Okay, this is the last one for today. Again, once you're finished with this video, you'll see a mitosis video for you to watch. Make sure that you watch that. Okay, so this is the last one for today, and then tomorrow you'll have a little bit more notes to finish. I know, as you notice, that they're not completely done. If you were not here last week for the notes, you will get them when I get back on Wednesday. Okay, so here's the metaphase. And you can see how it's changed from this point to this point. Look at, they're all lined up in that middle point. So the centrochromes are fully migrated to opposite poles. And the spindle apparatus is completely formed. And the nuclear membrane it's gone, right? It's been fully dissolved. The spindle fibers have lined up the duplicated chromosomes along that equatorial plate, so that each sister chromatid has duplicated chromosomes facing opposite poles. And the correct alignment at this stage helps to ensure that each new daughter cell will receive one sister chromatid and from each duplicated chromosome when the sister chromatids are separated. So it will guarantee that they get one red, one blue, one green, and the same on this side. And they both have a set on each side. At this stage, the chromosomes are thick and short, and they're condensed. Okay, there you can see the actual version of what they look like during the metaphase. Let me go back to that. It's kind of cool. You can see that the duplication from one side to the other. We also have another type of visual image. You can see how fat they are. Okay, so this is where we're going to stop. And at this point, put your notes away for tomorrow's video, and you will go watch the mitosis video with the Amoeba sisters. There's a slide deck that was put up today that you need to also be working on. I think the directions are on today's agenda. Okay, until tomorrow's video, bye.